Welcome everyone. We are going to start today. We start for the first time with the initiative Discovering the Nobel Laureates. In this annual event, we will bring you experts in the field to share their knowledge and help us to understand better the importance of the Nobel Prizes in science, mostly in physics and chemistry. But okay, before starting, who was Alfred Nobel? Okay, he was a remarkable human being. He was born in Stockholm in 1833, and he was fluent in different languages, like Russian, English, French. He wrote poetry and drama, and he was also interested in social and peace-related issues. He studied chemistry and technology, and he was considered as a radical during his life. Okay, today we have also a remarkable speaker. Her name is Christina Egestaval. She is the coordinator of the Cell Therapy, Stem Cells and Tissue Group from the Basque Center for Blood Transfusion and Human Tissues from Biocruces Vizcaya Health Research Institute. And she is also the coordinator of the expert group in stem cells in the European Society of Human Reproduction and Embryology. Before Christina starts, I want to remind you that if you want to ask something to her, you have a panel for the questions and answers, and Christina will answer your question at the end of her talk. And when you want, Christina, the floor is yours.
Okay, so good morning to all. First of all, I would like to thank BCMaterials for this really nice initiative, especially, well, Raquel, Unai and Senenchu, because, well, at the end of the day, we are collaborators in several projects. And, well, I will start to talk about this Nobel Prize of Chemistry 2020 that is focused on genetic scissors. Yes, now. So I'm going to divide my talk in these bullet points. First of all, to give you just a summary about who was winning, was awarded the Nobel Prize of Chemistry this year. What is, by definition, the genome editing, a little bit of history, how many tools we have, what is the mechanisms that is using these tools. Then I will give you an overview about the current clinical trials using these tools, especially in somatic cells or somatic cell therapy. Then I will, as a potential use in the future, the gene editing in the germline, and also stem cells, in stem cells, that I explained you briefly, one of the research line in my lab that is focused on correcting a specific point mutation in stem cells. Then there are two main issues to fix, to use this technology in order to be safe in the future. And there is a couple of societies or groups of experts that recommend through some guidelines the potential use of this gene editing technology, and I will conclude with future remarks. So, as you probably already know in the news, the Nobel Prize of Chemistry of this year was awarded to this CRISPR-Cas system for the discovery and development of a method for genome editing. And it's a big pleasure to introduce these two big researchers, women, because it's a pity in the Nobel Prize there is only 6% of women that was awarded through the history of the Nobel Prize, and this year is two women, Emmanuelle Carpentier and Jennifer Doudna, discovered this technology, the CRISPR-Cas genetic scissors, and with this technology the researchers can change the DNA of animals, plants or microorganisms with extremely high precision. And this technology has had a revolutionary impact, especially in some cancer therapies that I'm going to explain you later, and probably will treat or will cure or will correct some diseases in the future. And here below you see the couple of main publications from both researchers. Carpentier published in Nature 2011 that demonstrate a small RNA called track CRISPR RNA. It's a small guide that is needed for this CRISPR-Cas machinery that has to work. And Dovna, 1 year later in Science, was published that the Cas9 found in Streptococcus bacterial cooperates with a guide RNA that works like a scissor. So basically, this is the brief introduction about the two laureates of this Nobel Prize of Chemistry of this year. But it is true that I have to mention, because it's, I think, an honor to have, was Francisco Mochika, is a Spanish researcher from University of Alicante that also was nominated for this award, but unfortunately, he didn't get it, but his discoveries or his experiments previous to this, these two discoveries from Carpentier and Dovna were very important. Mochika described the complete gene sequence repeats in archaic organisms and was reported this CRISPR sequence in the first paper published, here you have it below, in 1993. And he was proposed the CRISPR acronym of this cluster regularly interspaced short palindromic repeats, because it's too long name for these repetitions of or repeats in this CRISPR system. And well, basically he published in 2005 the second paper, big paper. Unfortunately, tried to publish in big journals, but I mean there was some rejected in big journals, and finally was published 2 years later in this journal. So, as I mentioned before, the genome editing, by definition, is a type of genetic engineering in which a fragment of DNA is inserted, deleted, modified, or replaced in the genome of a living organism. So Nature Methods in 1911 was selected this genome editing as the method of the year, and a few years later, Science was selected the CRISPR-Cas system as the breakthrough of the year. So a little bit of history about the gene editing tools, because we don't have only a CRISPR-Cas system. We have also another nucleases. So the first nuclease, meganuclease, this is the first tool, was used in mouse cells in 1994. Then years later, in 2005, in human cells, the second nuclease, the same finger nucleases, was used for correction of a specific mutation. Then in 2011, the third tool is called Talents. Was edited some the genome of the human cells, and in that time up to 2013, the CRISPR-Cas was used in the human editing, and the Cas9 family of these proteins has, it's a huge family of proteins, and there is another one that is very promising. It's called Cas12, or also CPF1. That, we'll see if it's the new replacement or not of this Cas9. And surprisingly, 1 year ago, in October last year, there was a publication from American group that presents a new version of the Cas9 protein, and they call prime editing. So I'm going to explain you later what are the differences between all of them. So as I mentioned, we have this is the first tool, the most old one, let's say, meganuclease, thin finger nucleases, the transcription activator like effector nucleases, talents, and the CRISPR-Cas. So as you can see here, there are differences between them, but I think this table summarizes very well the differences in terms of a lot of criteria. For example, well, the winner or the gold standard or the best is the CRISPR-Cas, because you can see here is the table is in green. And the CRISPR-Cas is, let's say, is quite simple. The construction of the strategy. The in vitro testing also is very simple. The targeting efficiency is quite good in comparison with the other ones. This CRISPR-Cas permits to make more cuts or more corrections through the genome. So it's possible to make multiplexing corrections in comparison with the other ones that is not possible. And also the time and the cost is very low. So in general, it's the best one, but there is still one undesirable or unwanted effect of this technology that are called off-targets. What are the off-targets? Basically, you want to, for example, you want to correct a specific point mutation into the genome. Of course, you want to be more accurate in that specific region that you want to correct that mutation. But the off-targets means that it is possible to make cuts in another parts of the genome. And of course, this is not appropriate or accurate. So still there is a problem to fix with this technology in terms to avoid these off-target effects. So what is the origin of this CRISPR system? Well, basically, the CRISPR is a prokaryote adaptive immune system. Here in this cartoon is really nice cartoon that summarizes what's happened in a bacteria when it's infected by a virus. So to start with, when the virus is infecting the bacteria or archaea, there is this CRISPR array that is here, the spacer sequences of the CRISPR that are transcribed into this single or pre-CRISPR RNA. This is a kind of guide that is guiding these proteins, the Cas proteins that are the nucleases that are going to cleave and to cut the genome of the virus DNA. So it's quite simple, the mechanisms of this immune system of this bacteria. So the CRISPR-Cas system function, it is a defense mechanism to prevent repeated infection by the same virus. Let's say it's a kind of vaccine for these organisms. And the viruses cannot infect bacteria harboring homologous spacer sequencing, suggesting that these sequences play a role in the adaptive immune system in prokaryotes. So this is the origin of the CRISPR-Cas system. So the next question is, how are performing these Cas9, these cuts or these scissors are cutting in the genome? So any cell type or any cell in our body has DNA repair mechanisms to repair a specific point mutation, and the Cas9 is doing the same. So we have two pathways to DNA repair normally. So the first one is this one, the non-homologous enjoining, and the second one is the homology direct repair. So the first one, I have to say that is less precise than the second one. Why? Because with this mechanism repair, it is possible to introduce indels. What are the indels? Insertions or deletions unwanted. So, for example, and the second one is more precise, for example, in order to correct a specific mutation. So, if you want to correct a specific mutation in the genome, in a specific cell type or whatever, is much better to use the second mechanism or the second pathway. Or if you want to, for example, to make a knockdown in a specific locus in a gene, well, this is the first mechanism pathway is the best one. So, well, you know that from, let's say, 10 years ago or 8 years ago, basically this is a completely revolution in terms of publications about CRISPR in every single journal, and especially in the top ones, no? Nature, Science, Cell, but I mean, it is really interesting that there is a monographic journal only dedicated to CRISPR papers. So, well, here you have an example about the importance of this technology. So, as I mentioned before, 1 year ago, well, on the news, there was this discovery from this American group that they claim that with this prime editing, this new version of the Cas9, it is possible, maybe it is possible to correct almost 90% of the genetic defects. So now I'm trying to summarize the differences between the CRISPR-Cas9 versus the prime editing. So as simple as I can, because probably all the audience are coming from different, I don't know, different areas of the science. So, well, the CRISPR-Cas basically cuts the 2 strands of the DNA. So as you can see here. And as I mentioned before, this is, well, it is possible to create off targets. And of course, this is an undesirable effect of this technology. So, and one of the advantages of the CRISPR-Cas versus prime editing is that with the CRISPR-Cas, it is possible to get insertions or deletions of large DNA fragments, whereas the prime editing not. So, in principle, this prime editing is a more accurate tool than the CRISPR-Cas9, eliminating off targets, making this gene therapy safer in comparison with the CRISPR-Cas. So, and basically the mechanisms, how it's working, this prime editing is the Cas9 basically is a little bit modified in comparison with this one, the previous one. Basically it's a Cas9 modified and is cutting only in one of the two strands of the DNA, as you can see here. Also, there is a second enzyme, a reverse transcriptase called, reverse transcriptase, and guided by our RNA chain called pegRNA, as you can see here, and that is performing the edits and the cut site. So, and as I mentioned before, this group claimed that almost 90% of the DNA variants associated with diseases could be corrected with this prime editing. Well, my comment here is, well, we'll see in the future if this prime editing is safer as they claim, of course, and if it's working as better as they claim. So we'll see, probably coming soon papers about this technology. So, and now I will move into the potential applications of this gene editing technology. And I will start with the current clinical trials by using this CRISPR-Cas technology. So the first application that is already in clinical trials is the ex vivo somatic editing therapy. And what is that? So basically, we have a couple of examples. For example, patients with refractory or relapse hematological cancers, like, for example, lymphoma or leukemia. It is possible there is a novel immunotherapy called CAR T cells or CAR T therapies. Probably you already heard about this. Basically, is through a leukapheresis, it's possible to isolate the T cells from that patient with the leukemia. Then in a clean room, it is possible to introduce a chimeric protein. It's called chimeric antigen receptor, or CAR. And the way to introduce this CAR into the T cells, it is possible to introduce by lentiviral vectors or by CRISPR-Cas. So the FDA approved the first clinical trial to infuse those CAR T cells into a patient's. And this CAR was introduced by CRISPR-Cas. So this is one of the applications that is already in clinics. And the second application, well, the same application, I mean, but in another type of hematological disorders, CRISPR Therapeutics is a company, an American company that approved the EMA, the European Agency of Drugs, like the FDA, but by the European one, approved the first clinical trial to correct some mutations related with diseases like beta thalassemia and sickle cell disease. So here there is the first phase 1 or 2 clinical trial to treat these patients with beta thalassemia and sickle anemia. I remember last conference in Barcelona, previous to this COVID pandemic, obviously, was last year in Barcelona, the European Gene Therapy Society. This company presents the first preliminary data of these patients, and I remember that were very promising, a very good results. So we'll see the next patients and the next results. So here is the pipeline of these CRISPR therapeutics. As you can see here, they are developing with CRISPR-Cas different products, self-products, to treat these diseases, beta thalassemia, sickle anemia, all these CAR T products, and also other types of diseases. So another company that is involved, there is plenty of companies, but I mean, I cannot give you an overview of all the market because I mean, it's impossible to cover all of this, but I mean, Editas, this is another company that, as you can see here, there are different diseases that are, of course, developing these clinical trials. But I mean, I will be focused in the first one, liver congenital amaurosis 10, which is a degenerative disease related with the eye. And the second application of this technology is focused on in vivo somatic editing therapy. What does it mean? Basically is that you can, for example, you have a specific point mutation or mutation into a tissue or organ in your body. Then it is possible to inject in that tissue, for example, the machinery or all the components of the CRISPR-Cas in order to correct that defect and try to rescue the normal phenotype of that affected cell in vivo, not ex vivo as the previous application. So here you have a cartoon of all the steps of this clinical trial from Editas that basically they inject here this product that is having all the reagents of the CRISPR-Cas behind the eye in order to rescue the normal, to correct the mutation that is giving these defects in the photoreceptors in the eye, and to rescue and to correct the normal phenotype of these photoreceptors. So I just yesterday, I think, I read this news from Editas that they complete the first cohort of patients with this disease is a phase 1/2 of the clinical trial, but due to the COVID pandemic, of course, there is a little bit of a slowdown of the clinical trial. So, but I mean, we'll see. Hopefully, in a few months' time, the next results of this clinical trial. And the third potential gene editing application is in vitro germline editing medicine. So this, obviously, this is only now for research. There is any application in the germline, in embryos, in oocytes, in sperm, modifying some specific genes with this technology. No, nowadays it's only under research. But I'm going to give you an overview about what is the, let's say, the current status of these experiments in different types of cells in the germline. So we have these four, let's say four type scenarios in the germline. The first one is the embryo through the IVF or ICSI. It is possible to generate in vitro a human embryo. Then the oocyte, the female gametes, and also the male gametes, the spermatogonia stem cells, that could be correct some specific defects in the future in this germline. And also, there is a potential application by using gene editing in the iPS. The iPS are induced pluripotent stem cells that it is possible to generate a pluripotent stem cell line in the lab coming from a specific somatic cells, for example, a fibroblast in the skin. So was awarded Shinya Yamanaka, the professor Shinya Yamanaka was awarded by the Nobel Prize of Medicine 2012 for this discovery. I mean, to go from a fibroblast into a iPS, this process is called reprogramming. So, I mean, are having pluripotency features thanks to introduce 4 genes, the 4 Yamanaka factors, into this fibroblast. So once you have this iPS from a specific fibroblast, for example, in a patient that is having a mutation, and a disease related with a mutation, it is possible to correct, for example, that mutation and differentiate in vitro into the target cell type that you want, because the iPS are pluripotent and are able to generate in vitro, in the lab, any cell type. So ectoderm cells, endoderm cells, mesoderm cells, and also germ cells. So this is the potential applications of the CRISPR-Cas in the germline and stem cells. So to start with the embryos, before to start with the embryos, I want to give you this summary that, for example, a couple that has any, for example, any monogenic disease, is highly recommended to perform a FIP in order to have a baby. Why? Because there is this technology, this procedure, pre-implantation genetic diagnosis, PGD, that is to identify genetic defects within the embryo, and this is in order to transfer into the mother the healthy embryo and not the embryos that are carrying the specific disease. So this is a routinely technology or procedure that the IVF clinics are performing in a specific situation when one of the parents or both parents are carrying a specific mutations. And well, basically, this is there are three types of PGD. It's called PGT. PGT-A, PGT-M, and PGT-SR, and they are detecting aneuploidies in the embryo, monogenic disease in the embryo, and structural rearrangements in the embryo. So in this cartoon, you can see that when is the best moment to take a biopsy of this embryo, performed after FIP or ICSI in the lab, and normally at day 3 of the development is taken a biopsy and is analyzed this biopsy by sequencing if it's carrying or not a specific mutation. And as I mentioned before, the transfer, the embryo transfer into the mother is only make the healthy embryo, not the one that is carrying the mutation. But it is true, this PGD test is not covering all the situations related with, genetic mutations, diseases, or infertility problems. So maybe in the future, maybe because there is a huge debate about this, I'm going to tell you later, there are some potential uses of this genome editing in human embryos. For example, when both members affected with the same monogenic disease, maybe it's possible to use this CRISPR-Cas in the embryos. When one member homozygous for an autosomal dominant disease, maybe it's possible, and when one member affected by a chromosomal structural aberration could be use this technology. So we'll see. Again, the PGD is a procedure only to select the embryos, whereas the gene editing is to repair the embryo. So there is a completely different legislation about one or the other. So, I mean, in the future, this technology is using in the germline, a lot of changes has to be changed, of course. Yes, these are the main very few papers published with the use of the CRISPR-Cas in human embryos in order to correct some specific mutations. So I mentioned before that one of the side effects are the off-targets, but there is another one in the embryos only, the mosaicisms. What is the mosaicisms? I'm going to explain you now in this paper. Basically, up to this paper, published in Nature by Mitalipov in 2017, as you can see here in this cartoon, if you want to correct a specific heterozygote mutation in a gene, normally to perform that, you have here the zygote. Once it's fertilized, the oocyte. Here in the S phase, the CRISPR-Cas, the machinery of the CRISPR-Cas was injected in this stage, and finally, more or less 100 of the embryos at 8-cell stage were mosaic. This means that some of the cells within the embryo are corrected and other ones are not corrected. So this is, of course, this is not okay in order to correct the embryo. So, but this group, they were thinking that maybe if they inject the machinery of the CRISPR-Cas a little bit earlier in the early development of the human embryos, especially in this stage when the oocyte is fertilized with the sperm, then they introduce the CRISPR-Cas machinery in this specific moment, and they get finally this 100% correct all the cells within the embryo. So basically, they claim that the efficiency of this technology is around 45%, and these 45% of the embryos were no mosaics, and they don't detect any off targets. So, well, we'll see. Of course, there is not so many results or so many papers in this specific field because not all the countries around the world has permission to use human embryos for research, of course, but modifying with CRISPR-Cas. For example, talking about Europe, the first one that permits to use these embryos was UK, then Sweden, then Belgium, and this year, Spain too. So, well, we'll see if we can do relevant data in Spain, in our country, with this technology to improve this. Okay, this is the status of the correction of human embryos by using this technology. The second cell type, or the female gametes, are the oocytes. So basically, this data published by Juan Carlos Espizua Belmonte in 2015. So basically, there is very, very severe diseases, the mitochondrial diseases that are transmitted by the maternal mitochondrial DNA. And basically, they create this mixed model. By using mouse oocytes, they introduce a human mutated mitochondrial DNA. Here you can see here in red. And instead to use CRISPR-Cas, they use, because there were some problems with the CRISPR, they use TALEN, the third tool that I saw you at the beginning, and basically this they call mitoTALEN, and this mitoTALEN was eliminating the majority of the human mutated mitochondrial DNA. So, well, this is an in vitro model in order to think in the future to avoid this transmission of these mitochondrial diseases into the next generation. So, male gametes now. So the spermatogonia stem cells. So basically, there is not so many papers, so many data about the male modification, the male germ cells modification. But I give you an example. This is a mouse model that this model is having a specific point mutation related with the eye. And basically, they take out the spermatogonia stem cells, the stem cells into the testes. They correct that specific mutation by CRISPR-Cas9. They analyze later the off-targets analysis, the genotype, etc., etc. They put them back, those corrected spermatogonia stem cells, into a green mouse, into the testes. They differentiate those spermatogonia stem cells into spermatids, and they perform ICSI, a technology, ART technology. And finally, the offspring was completely healthy and corrected. So they claim that the efficiency of this technology in this mouse model, and of course in this type of cells, were 100 corrected and efficient. So, okay, we finished about the germline. And what about the stem cells? I explained you before what are the iPS and how we can generate in the labs the iPS. And well, it is possible to differentiate into any cell type. Okay, so one of the research line in my lab is about to model a specific rare disease, primary immunodeficiencies diseases that are related with defects with the immune system cells. In our case, with NK cells. And basically, the only curative treatments of these primary immunodeficiency diseases, the treatments available today is the hematopoietic stem cell transplantation. There is any other treatment available, and with this research, it is possible to understand the pathogenesis of these diseases, and maybe could be a future treatment based on NK cell immunotherapy by using iPS technology and the CRISPR-Cas technology. I explain you now. So we are focused basically in this one of these primary immunodeficiencies is called APDS2, that has a specific point mutation in this gene. And basically, thanks to the collaboration, so many people involved in this initiative, but especially the Hospital 12 de Octubre in Madrid. And basically, we had primary cells from the skin of the patient. We reprogrammed those cells into patient-specific iPS. Then we correct the iPS with CRISPR-Cas to rescue the normal phenotype of those cells. And of course, the target population of these deficiencies, the NK cells or the hematopoietic stem cells. And basically, we would like to differentiate this corrected iPS into the NK cells that are missing or are very low in the patient. So we just published, I think, well, 1 week ago, we generate the first line of this patient-specific iPS, and here this is the fibroblast, this is the colonies of the iPS, and this is the typical characterization of the iPS line. So basically, we have done the first objective of this research line, and now we are ongoing to correct the specific point mutation. Here you have the point mutation. We are going to use these mechanisms to repair this point mutation, and we are using hi-fi Cas9. We design my postdoc, obviously Marta, which is fantastic to do this fantastic work. I mean, the design of all the steps, or all the reagents, let's say, to perform this correction with the Cas9, the hi-fi Cas9, the guide RNA, and the template of the DNA. So, and the next step will be to differentiate those corrected iPS into the CD34 positive cells or NK cells that are related with the patient, that are genetically related and corrected with the patient. And we develop this protocol to differentiate from iPS into the CD34 positive cells and NK cells. And I'm not going to explain you the difference of this because it's a mess and I don't have time to do it now. But basically, we get really nice data about the progenitors from iPS, and the next part as well, we have data, but we need to perform more experiments. So basically this, as I mentioned before, there are some issues that still are technical problems that it has to fix. The mosaicisms in the embryos and the off-targets in any type of cells that it has to be to correct or to whatever you want to do. So to avoid mosaicisms in embryo, we saw in that publication by Mitalipov in Nature that performing or injecting the machinery of the CRISPR-Cas earlier in the early development, it is possible to avoid these mosaicisms. But also, in some specific cases, maybe it's much better if it's much better to modify the of one of the parents, or instead to modify the embryo. So, this is a couple of solutions to avoid this. And about the off-targets, we have tools to predict off-target sites by using different guide RNAs. Here you have a table with different links that, well, you can enter and give some information about the locus that you want to correct or whatever the strategy that you want. And they can give you a score about, well, which guide is the best one, and also where will be the off targets among the genome. So, and here in this other table, you have different strategies to minimizing these off targets. So, but because this is very complex, but I mean, to summarize this, it is very important, the delivery method of this Cas9 protein. And I think one of the best or most accurate to avoid these off targets is ribonucleoproteins delivery instead plasmids or lentivirus. So this one, the proteins is the best or is better nowadays. And also, depending of the type of the Cas9 that are using, for example, there is a high fidelity Cas9 is much better, is more accurate and more specific in the cuts in comparison with the Cas9. So, I mean, there is plenty of, combinations that, I mean, I give you, from my point of view, a couple of them that are fine to avoid these off targets. And about recommendations from a couple of societies, of committees. The first one is the National Academy of Science that published these recommendations or guideline or report about the potential use of genome editing, was published in 2017, and 1 year later, the NAFIL Council on Bioethics was published this report, more or less both of them the same. So here you have a traffic light that give you an example that they recommend, for example, the genome editing for basic research, including embryos. Why? Because it's needed to fix these off-targets problems, the mosaicisms in the embryos, and a lot of small issues in the technology, that in order to have this technology as safer as we can, no? So they claim that this is a huge recommendation. And also for somatic gene editing to treat serious diseases. We already saw in clinical trials this CAR-T application in cancer patients, and these hematological disorders, for example. But, of course, it is not recommended, for example, here in red, somatic or germline editing for enhancement purposes. So this is completely forbidden. And I give you now an example. So just 2 years ago, there was a huge meeting in China about the human genome editing. And this researcher, the He Jiankui, was presenting the first data. He present that he was the first who was doing human genetically edited babies, were twin girls, Lulu and Nana, and basically it was completely a scandal, and everybody in the audience was in shock. Basically, the parents of those babies were HIV positive, and there is a mutation related with the resistance of HIV infection. And basically, it's this one, CCR5 delta 32, and this Chinese researcher was induced this specific point mutation in those embryos. And well, in order to prevent a future HIV infection. So this is unbelievable. So all the societies, genetics experts on the field were condemned this. So, and 3 years later, the Chinese government decided to go into detail this Chinese research for at least 3 years. So it's not possible to do whatever you want especially in the germline, because I mean, this is a big issue. So, well, it was a completely scandal. Maybe you already know by news or on TV or whatever. So, and last year was published in Nature News as well, this Russian embryologist that wants was also to CRISPR edit babies. Anyway, so this is a little bit, I don't know, worried about these people that wants to, well, to do these things without a general consensus, and also because there are some specific issues in the technology that has to be fixed. And well, the World Health Organization committee was recommended that there has to be a registry for all the studies that involve editing the human genome. So, well, and just to finish, some conclusions and future remarks. So, somatic cell therapies with CRISPR-Cas9 are getting promising results, as you saw before, CAR-T, beta thalassemia, to correct those diseases. The genome editing cannot be actually performed with sufficient precision to allow scientists to creating genetically modified babies and germline cells, because there is an accurate editing, the mosaicism is still there, and off-targets mutation as well. And the big question is, the prime editing could be the new and safe tool in comparison with the Cas9. So, well, we'll see. And of course, the research in the human germline, gametes, embryos, and also the stem cells should be promoted to determine the usefulness and safety of the technique, and also is an excellent tool, as I mentioned before, for the generation and to study diseases model, and as well for the study of genes involving in pre and post-implantation development and gametogenesis. And still there is a existing open debate worldwide, and a general consensus should be established in order to use the gene editing technology, especially in the germline in the future, if it's the case. So we'll see. So, and thanks for your attention.
Thank you so much, Christina, for your very, very nice talk. I think that they are very promising results, and clearly this technique has a high impact on our society, of course. Now, if you agree, you can start answering some questions that we have here, some of them. Wait a little bit. Yeah, I have this view. Okay, the first one is about where those human embryos used in these papers are coming from.
Yes, well, basically, as I mentioned before, not all the countries around the world has permission to use stem cells derived from human embryos, and these cells are embryonic stem cells, and also to use human embryos for research. This is to start with the permission in your country, and then, for example, the couples that are going to IVF clinics, and for example, they get, for example, 7 embryos, and they are going to use only for make embryo transfer, only 2 or 3. The remaining embryos, they sign a consent inform in order to donate those embryos for research. So the origin of these human embryos for research purposes are coming for couples that has permission to use those embryos for research purposes.
Okay, thank you so much. Another question. This is from Wansun. Are there people still study to improve other gene editing methods, such as zinc finger protein?
Yes. So I'm focused more in CRISPR-Cas because, well, at the end of the day, the Nobel Prize was awarded and is the most used, let's say. And as I mentioned before, there is the gold standard. But yes, of course, there are a lot of papers and a lot of groups around the world that, for example, are improving other tools like thin finger nucleases or proteins to use this. And especially, for example, I give you the example, in the oocyte, the CRISPR-Cas is not working well. Due to, well, I'm not going to explain you, it's a little bit, if you read that paper, you will understand. But anyway, and in that paper, for example, or in this cell site, in the oocyte, is working better, the talents, instead the CRISPR-Cas. And probably the thin finger nucleases in some specific situation or cell type, maybe is working better, the thin finger nucleases in comparison with the other one. So, yeah.
Okay, thank you. And we have another question. What is your personal view on the DNA modification in embryos? Is it safe for future from your point of view?
So, okay, this is the $1 million questions, and it's as usual difficult answer to tell you. But nowadays, my point of view about the DNA modification in human embryos is not ready yet. So, I mean, as I mentioned before, there is a lot of issues to fix before to claim that is safe technology in order to some specific defects to correct in the embryo, because also we have this PGD technology that, I mean, is covering a lot of diseases or mutations or problems related with infertility that is working fine and is not modifying the genome of the embryo. And in the future, imagine the hypothetical situation that, I mean, everything is working perfect, is safe enough, etc., etc. Honestly, it has to be a general worldwide consensus to see if it's needed or not to correct some specific mutation or disease, and also has to be approved by I don't know how many committees, experts, ethical committees, blah, 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 etc. So I think it's a far away use from my point of view in human embryos.
Okay, thank you, Christina. And I think that the last question, because we are here almost 1 hour, Unai wants to congratulate you for the super clear presentation. And are there trials to correct genetic diseases in the adult, or is the technology limited to embryos?
Sorry. What is the? I didn't.
Yes, are there trials to correct genetic diseases in the adults, or is it the technology limited to embryos?
No. Yes. You mean in the somatic gene therapy? Yes, is already in clinical trials, as I mentioned before, to correct in adults. Of course, yes, and in embryos only is just related for research nowadays. So I answer correctly?
Yes, I think so.
Okay,
yes,
fine.
Yes, I think so. Okay, thank you, Christina. I think that we are going to stop here. Thanks for your very, very nice presentation. We learned a lot, I think, and it's an amazing topic. And okay, thanks everybody for attend to this series of discovering the Nobel laureates. We hope see you in the next one, that is the physics prize. Okay, thank you so much.
Thank you.
Bye.
Take care. Bye bye.